So one of the things about the transport, guys, is like the water potential stuff, guys, and that's I can't say it's a big thing, guys, but by the way, but that's one of the stuff that you have to know. Definitely the homework that we talked about, guys, by the way. I mean, the recording is online as well for the AP, so you can go back and look at it. That's sufficient enough to solve questions in homework as well. Definitely on the AP as well. So the other stuff, I mean, again, very targeted for some single couple questions here and there. So when you guys do the homework, then I'll make some comments. You know, when we go over the homework, guys, by the way. So, but today, guys, like I really want to go ahead and start the cellular respiration chapter, the cellular respiration. So this chapter, guys, respiration. Let's talk about the overall picture. Okay. So we talked about it, guys, before, where if glucose molecule is C6H6 in terms of monosaccharide equation, right? So we kind of talked about it where these, what we call the monosaccharides or sugar, they provide energy, right? So that's what we talked about. So I've never mentioned to you guys where the energy is in, right? That's one of the stuff we talked about. So based on the discussion, guys, I said, okay, so energy is in the electron, okay, in the carbon electrons. So the electron, the valence electron of the carbon, that's where the energy is stored. That's one of the things that we talked about. Okay. So if you count the number of electrons overall, so let's do some counting here. Okay. So if you count the total number of electrons in the carbon, that's a valence. There's 6 of them. So it becomes what, guys? Like minus, like I mean, so there are 24 electrons overall. So each glucose will provide 24 electrons. That's what they call, guys, high energy electrons, right? So let's kind of think about, guys, what's really happening here. So in terms of the equation perspective, so you've probably seen the overall high in the equation, high level equation where this is they call it oxidation of the glucose, right? Turns into CO2 and H2O. So I'm not going to balance it, by the way. So there's a 6H2O, CO2 and 6H2O. So I think that's a balanced equation just for the molecule perspective here. So I don't care about the oxygen right now. I just care about the carbon right now. So let's take a look at it, guys, by the way. So you guys are coming from the AP Chem, so you guys know how to get the oxidation state, right? So like charges, right? So if you look at it, guys, oxygen is -2, so it becomes what? -12 for the whole charges. This is +1, so +12 for the whole charges. The charge of the carbon would be 0. Okay. So if you look at it, guys, you guys have to know to do this, guys. Carbon's charge will be 0 for the glucose molecule, which means what? It hasn't really lost or gained. That's why it still has that 24 valence electrons. But now, if you look at this guy, this carbon right here, what happens, guys? This carbon is what, guys? Charge will be +4. Okay, here is 0, that's a +4, which means what? Every carbon, they lost 4 electrons overall. Which means what? Okay, so all the electrons they had is gone, completely just oxidized, okay? Then where did it go? It went to what? It probably went to the oxygen. So the last stop of the electron is the oxygen. Okay, oxygen is what they know to be called what? The last electron acceptor of the whole process. Okay, so all the electron, you know, all went into the O2 gases, okay? Which in turn they turned into what, guys? CO2 and some of the H2O together. So that's what's really happening, guys. So the electron goes from the high level perspective, okay, electron goes from carbon to the oxygen, okay, and then they release energy. So how do they do this? How do they freaking do this, guys? Yes, how do they provide the energy for them to produce ATP, right? So one thing to understand, guys, is this, okay? So on a very high level, guys, so in terms of understanding of the oxygen is very electronegative, okay, very electronegative, okay, which means that they pull the electron to itself, as you know from AP Chem perspective as well. You know, they have the highest, second highest electronegativity, and also oxygen has a very high electron affinity as well. So if they gain electron, they release a lot of energy. So, so basically, guys, understanding. So you guys remember this, right? Like you just took a test. I just remember you guys were talking about the AP Chem here. So the principle is right. So this is N1, N2, N3, N4, you know, N5, N6, like whatever, you know. So as we know, guys, if electron drops from the higher level to a lower level, okay? Yeah, what the hell happens, guys? Yeah, what happens, Andy? Yeah, what happens, Andy? So electron from the higher level to lower, what happens?
It's released. Energy is released.
Energy releases, exactly, okay? Because why? Potential energy electron is given away, okay? As what? Like, yeah, like photons, right? Like you guys heard it, right? So this is what? The principal energy level. So I'm not gonna teach you chem right now, by the way. But now, same thing kind of applies, you know, with the process, if you think about it, right? It's because carbon doesn't have that strong of affinity of electrons, okay? But oxygens do, okay? So now, what it means is, if electron is gained by the oxygen, okay, what happens, guys? Electrons are more stable, okay? Because electrons are attracted more to the oxygen. So what I'm saying is, okay? So electron here represents what? Electron, the carbon, okay? When the electron is in the carbon, that's the potential energy of electron, okay? But now when it gets to the oxygen, it's what? It's dropped down all the way to the bottom. Okay, this is when oxygen gains electron is what? When the potential energy, in bio they don't talk about as free energy, goes all the way down. Okay, so which means what? Because carbon, when the electron is in the carbon, this is how much energy they have. Because it's kind of loosely bound. Carbons, they do not have that high electronegativity values. But now, the electron is highly like energetic. But now, if it drops down to the oxygen, what happens, guys? They lose it all. Okay. For example, guys, when you're little kids, your mom and dad probably, yeah, go play, you know, you're all over the place, right? But now, like, oh, yeah, you guys, college is a minute now, so you guys have to study, like, you're kind of strapped down and study, so you can't really be able to move around, and it's the same thing here, okay? Carbons don't have that strong affinity with electrons, which means they're really high up in the free energy perspective, but oxygen isn't. That's why so when carbon gives away the electron to the oxygen, happens, guys, this much energy is what? Yeah, like, it should be released, right? So, here's what cell is doing is what, guys? They capture this transition energy and what? Convert it to what, guys? ATP. Somehow they convert to ATP, okay? That's the core of the today's chapter. How do they do this? Okay. Okay, so let's jump right in, because this is the overall process, by the way, but that's basically where it does come from. So one of the stuff that we talked about is, you know, okay, so we know that there is something we call lipids, right? So lipid, guys, like for example, they have a very long fatty acid chain, guys, triacylglycerol, you know, that's one of the stuff they've talked about. So, you know, like, just for example, guys, like C, like, you know, C like 20, like H like 50, like O2, like for example, like, you know, that's basically what the formula for lipid is typically, because they're, I mean, they have the same carbon, hydrogen, oxygen, but there's a ratio is different, right? There's a lot more carbon versus the oxygen, right? Which means overall, by mass percent perspective, there's a greater number of carbons, right? Which means what? Because of this, what happens, guys? Carbons have electrons, okay? Because of that, what happens, guys? They can provide more electrons for energy perspective, okay? For example, you heard it, right? Like, dude, if you guys have 1 gram of this guy, 1 gram of that guy, which one can give you more energy? Of course, why? It was this one. Because why? Now you should know. Because why? It's because what? They got more electrons in there. Okay, so let's talk about the first process that occurs in this overall, you know, the whole picture perspective, guys. Here. So, this is the first process called glycolysis. Okay. Glycolysis. Okay. Glycolysis. So glyco means sugar, lysis means splitting, so splitting sugar, okay? Very simple. So your teacher will say glycolysis splits sugar, you know, sugar goes in, okay, and then splits into pyruvates, and that's it, game over. The next question. Not that easy anymore for you guys, okay? Yeah, you gotta understand, guys, okay? I don't even like I gotta go back and like remind myself, because like it's not something you can just put into your brain and have it like, you know, just ready every time. Okay, so the 10 steps of this, guys, is just I don't know, you guys have to, I don't know, constantly remind yourself, guys. By the way, but what I can tell you is this, okay? You guys are like, what do you guys want to do, guys? Like, you want to go to med school or something? Is that what you're gonna do? Medical school, guys? Yeah? No? What about you? What about you?
Yeah.
So, you see, when you go to med school, right? I'm recording this, okay? You guys have to take the class called MCAT. Do you think you have to memorize this? What do you think? Absolutely not. Okay, they don't want to talk about it. Okay, they don't want to memorize. So, you guys understand the difficulty of this exam, okay? It's not difficult because the material is like super hard, but it's just that like how much you have to retain. That's where it comes down to, okay? And then like you guys come and talk it up, guys, and I'm just kidding. You guys have to, like, when I see, like, dude, like, just for today, like for starting from like Monday night, guys, okay? Like so I don't know if you can see it, guys, okay? I made how many pages of notes, guys, here? I guess So, dude, like four or five pages, guys. I mean, I have to constantly write it down. But I mean, like, or are you guys spending that much? I'm not taking the test, guys. By the way, okay? You guys are. So, you know, I can just tell you, oh yeah, memorize this. Next chapter, I can do that, okay? Just so you know. All right, so glycolysis, by the way, is, you know, glucose, okay? Glucose comes in, okay? So the way that glucose comes in, I mean, I don't want to talk about it, guys. I mean, how the glucose comes in, right? I mean, so transport, right? So how can glucose come in, guys? So this is one of the few things that we talked about, okay? So towards the end of last class, we talked about how glucose, when it gets digested by the, you know, by the, like, what do you call that, guys? In the digestive system, right? How do the glucose come into a cell, guys, by the way? How do they come in? They can come in as what we call the co-transport. Okay, I mean co-transport. So co-transport, guy, I mean, you guys go back to the recording, guys. You guys can listen to it, by the way. So they pump out the Na pluses. Na plus will hook up with the glucose, and comes back in. Okay, following the concentration gradient of Na plus, you know, stuff like that we talked about. So glucose comes in. Okay, whatever the glucose came into the cell. Okay, so glucose can come in to the epithelial cell of the intestine, by the way. But I mean, that's a whole different different chapter to discuss on that, by the way, human physiology. But again, let's say glucose come into a cell, any cell, okay? So what happens? The first step that occurs, guys, glycolysis. Glycolysis can be performed by any cell that we have, okay? So glucose can be split up as a glycolysis for the eukaryotic cell and also prokaryotic cells. So glycolysis is a very preserved process throughout evolution, okay? So evolution, guys, by the way, so meaning, I mean, we don't look like, you know, some of the counterparts, guys, right now, like some of the, you know, other animals that's primitive, but the process, such as glycolysis, is performed by every cell that's out there, okay? So what that happens here, so you might want to know the steps of this, okay, what really happens. So even like I don't memorize, like I remember the enzymes, okay, so I review them, but I don't remember like every product of the whatever, okay. So I mean, so this is something you might want to memorize, guys. I can't recommend to memorize like on the day, like, you know, like today, but ultimately before the test, guy, you have to memorize this, by the way, just so you know. But the problem is, if you don't memorize it early enough, you're gonna constantly get questions wrong on the practice problems. So glucose goes in, okay, so this is glucose. I mean, it's better to look at the, I guess, your reading, guys, by the way, okay? If you guys have it out, guys, by the way. So glucose, guys, the first step, okay, there's an enzyme called hexokinase. So you have to understand what kinase represent, okay? Hexo means like in a carbon with the 6 carbons. Sugar with 6 carbons, by the way, okay? Hexokinase. So you need to understand what kinase, function of kinase. I might have mentioned it, by the way. The job of the kinase is what, guys? They actually, their job is, okay, to take a phosphate and put it into something else, put it on something else. That's their job, okay? So they sort of what? Transfer a phosphate from one party to the other, okay? So basically, guys, I'm gonna break this in 2 portions, okay? So the first half of the process, there are 10 steps, the first 5 steps. This is where, they call it energy investment, okay? It's called, guys, like where they invest energy investment phase of the glycolysis. Means energy is actually being put in, okay? So glycolysis is there for them to get ATP from glucose, but now they need some energy put in for the process first, okay? Just like, for example, guys, if you're gonna run a business, you gotta put some money down, right? You gotta put some money in there, you know, but that's basically what it is. So basically what happens is, okay, hexokinase is what, guys? So they have ATP. So they take the phosphate, one of the phosphate group out from ATP and what? Put into glucose, okay? Which means it will turn into, you know, like glucose with the phosphate. Okay, so now glucose has a phosphate group on them, okay? Because of what? The kinase. And then basically what we'll have after this, guys, will be there's an enzyme called phosphogluco isomerase. Okay. So I mean, again, it's something that I mean, even if I write it, guys, you have to memorize down the road. Okay. So basically, what you understand is that so phosphoglucosimerase is what basically what they're doing is turn the glucose into a fructose. Okay. So basically, the end product of this is a fructose. Okay. With a phosphate group. And the next step, guys, you got step 3. Is it's a phosphofructokinase. Okay. So it's kinase again. So you can kind of go from the reading if I make a mistake. Right? I mean, it is what it is right now. So which means they phosphated again. They're phosphorylated again. Okay, which means the end product is it's a diphosphate. So the phosphate is on both sides of the fructose. Okay. That's just third step. So still like, dude, this is imagine this is what at the expense of ATP. Okay. So what do they do, guys? They already use what 2 ATP for this. Okay. So they use what like okay. So 2 phosphorylated, which means what they use 2 ATP. Okay. So after this, so if you do the reading, guys, if you have done the reading, guys, then the term glycolysis it really comes from the 4th step. Okay, 4th step. Okay. The 4th step. Okay, it's by aldolase. Okay. So the 4th step, guys, basically what it does is it splits up. Okay, it splits up fructose in half. Okay, fructose is a 6 carbon molecule, but now aldolase, I mean, it splits it up in half. So now you have what? So I forgot the one of the guy actually, but now you get 2 products. Okay, it's called G3P. This is very important. Okay. So these are. So I forgot. This is like I forgot what this guy is, by the way. They're basically they're isomers each other, by the way. Okay, so G3P, by the way, okay, it's called glyceraldehyde 3-phosphate. Okay, glyceraldehyde 3-phosphate. Okay, it's called the G3P. Okay. So basically, guys, but this is basically you gotta understand, guys. Okay. Glyceraldehyde 3-phosphate. This is the end product of the photosynthesis. Okay, you guys probably heard, guys, photosynthesis. Okay, the plants make sugar, right? That is the end product of the Calvin cycle of the photosynthesis, by the way. Okay, so but now that's what they get. Okay, so this is basically, guys, what they can make out of the energy investments up until here is what they call energy investment. Okay, which means like they put a lot of ATPs in there. Okay, and now. This right here, the enzyme, this is the enzyme called the isomerase, okay, where they can, you know, shift from one of the isomer form of G3P into this guy, okay? So now, if you think about it, guys, by the way, you know, so then, okay, so if you produce G3P as the plant, how do they produce glucose? You what? Backtrack it, okay? Go backward. If you think about it, right? So now this is energy investment. So like, where's ATP? ATP didn't come out yet, okay? So, and also you can think about it also as well, right? So okay, so the whole purpose is ATP, but okay, what about the electrons? You know, so have we done anything with the electrons here? You know, that's one of the things you can think about as well. So, but now the step 6, this is step 5 here. So step 6, okay, step 6 and on. Okay, so this becomes what they call the energy payoff. Okay, so now they're getting paid. So the glyceraldehyde 3-phosphate, guys. I mean, this will. So this one, guys. So I forget exactly the name of this, okay, but it's the dehydrogenase. Okay. So the enzyme is called dehydrogenase. So something. So they always have a name, guys. By the way, so something like maybe glyceraldehyde 3, like dehydrogenase or something. So this is basically really important. You gotta understand what happens with these dehydrogenase. Okay. So these are the guys that what, guys? These are the guys that actually pulls the electrons. Okay. So basically, they're taking the electron out from this guy. Okay. And dehydrogenase basically is what they do is that they take the electron out, they remove the hydrogen, take the electron out from this guy, and they will reduce, okay, NAD plus, okay. So this guy, so NAD plus, okay, so it's nicotinamide amide adenine dinucleotide. That's what it stands for, by the way. I don't think you have to know that, okay, by the way. Okay, nicotinamide adenine dinucleotide. Okay, that's what this is, NAD. So basically what happens is so this guy is taking the electron out from this one, okay, 2 electrons, and then turn it into NADH, okay. So that's basically what it is, guys, by the way. So dehydrogenase, that's their job, okay, so you know. All right, that's their job. So, just go back to, yeah, I'm sure I'm not making a mistake. So the product there, I mean, so again, and also because, I mean, so keep in mind, guys, just so you know, there are 2 of these guys, by the way. So one thing to understand, guys, there are 2 of these guys, okay? Because this is a 3-carbon molecule. That's a 3-carbon molecule, okay? So they're technically speaking, they can have a 2G3 piece, okay? Nowadays, so which means they can do this process, and now which means overall they can produce what? 2 of these guys, okay? And also using the energy of this as well, there's no enzyme for this, but they'll phosphorylate this guy. So there's 1 phosphate group on them right now, but they'll phosphorylate also again. So basically the product of this is basically it's a 1,3 biphosphate glyceride. So end product of this, guys, is a 1,3 biphosphate glyceride. So that's the end product, like not really end, but that's the product of the step 6, okay? So basically what it is that there are 3-carbon molecule, okay, they're 3-carbon molecule, there's a glycerate, and they got what, 2 phosphates on them, okay. So I have seen questions about like this, okay, well I'll talk about it a little more, guys, but so keep in mind, guys, this step, step 6, right, it will not occur, okay, okay, if NAD plus runs out, okay? NAD plus must be in presence in order for step 6 to occur, okay? So reason for that, guys, bio, they love to talk about, okay, something they call this what, guys? A coupled reaction. A coupled reaction can be like freaking anything, okay, in bio. So, but a lot of times, guys, like coupled reaction can be what? A coupled reaction typically is what, guys? Is what? Reduction, oxidation reaction, okay, typically, right? So glycerol 3-phosphate, okay? And if you look at what the hell is happening, guys, by the way, okay? So you have an aldehyde, okay? You're turning into something else, so that's an oxidation, okay? So when you're doing oxidation, okay, then what must be coupled with it? A reduction process, right? So without a reduction, okay, you can't have an oxidation process. So without NAD plus present, okay, which means this is the one that gets reduced, while this is the one that gets oxidized, okay, which means what? It's not gonna happen, okay? So NAD plus must be present, okay? Nicotinamide, adenine, nucleotide, they must be present in order for this process to happen, okay? Otherwise, guys, if you run out of this guy, what happens, guys? It's not gonna happen, okay? So that's one of the things, guys, by the way, okay? So you can kind of see where this is going. Yep.
Is the enzyme just called dehydrogenase?
So there's a name for this. Dehydrogenase, there's a name. So what's this? They have a triose phosphate dehydrogenase, okay?
Do we need to memorize that one?
Yeah, of course, you have to know everything. So, dude, I'm not taking a test, but you have to memorize everything. So, yeah, so you gotta memorize the enzymes, okay? Make sure you gotta know your enzymes, guys, by the way.
How do we spell it? Or can we just memorize it?
It's in the reading, okay? So triose phosphate dehydrogenase, okay? Do the reading. Somebody read this, guys, okay? So here now, okay, after you produce that, so you guys have 1,3 biphosphate glycerate, okay? So what do you think will happen, guys? What do you think will happen? There's a phosphate group. So they gotta pay off. They gotta get some energy, right? Which means what? There must be now a kinase, okay? So now, from a 6 down, okay? 6 down, there's a step 7, right? Step 7, guys. So there is an enzyme called. So probably they will say. Let me see. There's a phosphoglycerol kinase, okay? Okay. Phosphoglycerol kinase. Okay. So, what do you think will happen, guys? What do you think? Kinase. What is a kinase? Is what, guys? They move phosphate groups, right? They take one of the phosphate group out, okay, and what? Produce ATP. So basically, what? This will produce an ATP now. So during the energy investment, they use 2 ATPs, okay? And now, like, they're getting ATP here. Plus, I guess they took one of the phosphate out, so 3 phosphoglycerate, okay, plus. So if you have taken, guys, like organic chem, okay, then I can be a little bit more explanative with why this is an oxidation, guys, but I'm not gonna go there, okay? And then from here, 3-phosphoglycerate, okay, because now they got only 1 phosphate. This is gone now, right? 1 phosphate group. So that, I mean, if I'm not mistaken, guys, okay, this is a step 8. So I know that there's an enzyme called a mutase, okay? I don't think this is a good, so it's the phosphoglycerol, so it's a phosphoglycerol mutase, okay? So basically what it does is, I mean, it's just, it doesn't really do much, guys. Mutase means it just changes the molecule a little bit, and then the product becomes, it becomes, guys, a 2-phosphoglycerate, okay? So I'm not drawing the structure, guys, by the way. I don't think you have to know the structures of these guys, okay? But you have to know the steps. It's better to memorize the steps, guys, okay? It's not, if you, like, I don't know, like, I don't have to teach you guys how to memorize the steps of the process and memorize them, okay? And then, step 9, I think, it's enolase, okay? So enolase, guys, it will turn into you probably heard this, guys, a phosphenolpyruvate, okay? So keep in mind, guys, I mean, they still got these, one of the phosphate groups still there, just so you know, they haven't taken that out. And then step 10, this is, I think this is also one of the kinase as well. I think this is a pyruvate kinase, if I'm not mistaken, guys, by the way. So the resultant is what? You get pyruvate here at the end. That's the end of it, okay? That's a pyruvate, guys, that's the end of it. So that's a 10-step, guys. You can definitely, with the ATP, okay? So keep in mind, guys, by the way, so there are two of these guys, by the way. Okay, there are 2 of these, because they separated here, guys, which means you're gonna get 2 ATP here overall, and you're gonna get 2 ATP here overall, because there are 2 of the G3P has been created, by the way. So overall process, so let's look at the high-end perspective, guys, okay? Glucose went in. Okay, 1 glucose went in. So you understand, guys, what came out of it. 1 glucose went in, we can put the other stuff if you want to, okay? 1 glucose went in, okay, and also 2 ATP went in, and also 2 NAD plus went in, okay, into the glycolysis process, right? And what came out of it is what? 4 ATP plus 2 NADH, okay, plus 2 pyruvates. So I mean, again, if you want to get into like nitty-gritty stuff, guys, by the way, then you gotta add, okay, so we added 2 H pluses there, okay, and also we added what, guys? I mean, so we added, you know, like ADPs, okay, 2 ADPs, and the 2 phosphate, okay, molecules, the 2 phosphate molecules here. So I mean, you're gonna get into nitty-gritty stuff, okay? That's what's happening. That's the overall process. So you're getting this. You got these right now, okay? And now prokaryotic cells, this is it. That's the word so keep in mind, guys, prokaryotic cells, this is where it stops, okay, so you know. So these 10 steps, okay, even very simple bacterial, like prokaryotic cells can do this as well, okay? And this is what the final product will be, 4 ATP. So they use 2 ATP, okay, which means, but yeah, they got like 4 ATP here, okay, which means a net amount of what, 2 ATP And then what? This guy. So probably don't really know what it is. NADH, guys, is what? It's known to be what we call the electron carriers, by the way. And then you got pyruvate. So also keep in mind, guys, pyruvate is not much different from the pyruvic acid. So some, I think you'd be mostly be getting the pyruvate. Okay, sometimes the questions we give the pyruvic acid, they're the same thing. Because there's no difference between these two. So there's the structure of this one, at least not know the structure of the pyruvate. 3-carbon molecule. 다 나와. Carbon, carbon, carbon. This is pyruvate. You understand, guys? That's a pyruvate. 3-carbon molecule, okay? And think about it, guys. This is a pyruvic acid, okay? Then what is a pyruvate? When it becomes what? Bronsted or conjugate base, right? That's a pyruvate. And the H plus popped out, right? So okay, is it truly different now? It's like, dude, which one do you think you found more of? Could it be more on the pyruvic acid portion, okay? Because why? It's a weak acid, okay? So a lot of places call pyruvates, by the way. So I was like, whatever, okay? But they're about the same thing, guys. You don't have to be concerned too much about it. So you gotta understand, okay? Yeah, what is this again? This is an acid. You know what acid? Do you see, guys? This is an acid. Yeah, well, what's glucose? What's glucose, guys? Glucose is what, guys? Glucose is what? What's an alcohol? It has an alcohol portion, right? It has a lot of OH groups on it. Okay? So I'm not talking about the organic chem right now, guys, by the way. Alcohol into an acid, that is an oxidation process. Okay, where did the electron go? They need what, guys? And so this is a couple reaction between what? Glucose into a pyruvate, and also what? NAD plus into NADH. Because what? There's a reduction process because glucose is being oxidized, okay? And for some of the prokaryotic cells, this 2-ATP, net amount of 2-ATP got, that's it, okay? So let's talk in terms of these prokaryotic cells right now, okay? For them, okay, they can run this process all over again, okay? When next glucose comes in, they can run the process again, okay? Because this happens in cytoplasm of the cells, okay? They can run this all over again. They got how many enzymes they got, guys, by the way? So remember we talked about it, guys, but just a review perspective. These enzymes, what, guys? These enzymes reproduce, even for the eukaryotic cells, the enzyme, like these enzymes, right? What does it say, guys? Dehydrogenase, okay? Phosphoglycerol. Yeah, you get what these enzymes, right? Let me ask you a question, guys, okay? Production of these enzymes should occur where? Where should they produce this, Andy?
Ribosome.
Production of the ribosome. Right? Where ribosome has two locations, as you mentioned last week, right? Which is one of them is ER and cytoplasmic. Which one?
Cytoplasmic.
Why?
Because it's used in the cytoplasm.
Okay. Okay. So, so if you look at it, guys, the problem with this, okay? So if there's a constant supply of glucose, okay, and constant amount of enzymes, this process should really be a problem, right? Yeah, 근데 what do you think the big problem is here? What did I say? 문제가 뭐야? What's a problem, okay, to run this process? What is the problem, guys? Yeah, what is the problem of rerunning the process? What is so? Think about it, guys. This is a product. The whole purpose is to produce ATP. So ATP will be doing a lot of what? So why is ATP used, guys? Because what? It's used to run some of the cellular function or process or reaction, right? We talked about that. Because why? If a reaction, okay, if a substrate gets a phosphate group, okay, they become more unstable, it increases their free energy for the run the process, right? So I mean, what we'll talk about, guys. So ATP being a crucial process or portion, right, of the reaction that is relatively non-spontaneous, okay, which is endergonic. So ATP is making it impossible for them, right? But now, yeah, so ATP will definitely be used, okay, turning back to ADP. Okay, ADP will produce, okay? So, and this is not a problem, the phosphate group is not a problem, guys, by the way, also as well. But now, what is the problem? What's the problem, guys? This is a problem. You see that, guys? NADH. Because why? They need to put it back into this guy. You know what I'm saying? Where are they going to get this from? Without this, an NAD plus, what happens, guys? the step 6 is not going to happen. Or step 6 is not going to happen. Okay? Starting from step 6, that's energy payoff. Okay? So which means they're going to use ATP, but what? This step is what? Blocked. You know what I'm saying, guys here? So yeah, what the hell? The purpose of this whole process is gone. Because what? Dude, you're putting glucose in to get an ATP, but if you don't have an ATP plus, what happens, guys? It's not going to happen. The process is not going to occur. You know what I'm saying? So what do they have to, guys? So if you are a prokaryotic cell, what happens, guys? You gotta figure a way out to what? Convert this back into an ATP plus. You know what I'm saying? Okay? If your only process of getting ATP is the glycolysis process, right? Yeah, what do you have to do? You gotta turn this guy into that guy, okay? What do you call it? What do we call that, guys? That is what they call fermentation. You know what I'm saying? That's what they call fermentation process. So there's a two, right? You guys have to know which is lactic acid and what? What so? Lactic acid with ethyl alcohol fermentation, right? So listen carefully, okay? So like you got this guy, okay? Pyruvic acid. Okay. You gotta understand, okay? Listen very carefully. You're turning this into what, guys? Ethyl alcohol. Where's the other carbon, guys? CO2. So, yeah, what did I say? Yeah. Alcohol to an acid. What? Oxidation, right? Acid back to an alcohol. What is that, guys? It's a reduction. Do you know what I'm saying here? So this is what? This is a reduction process. Yeah, think about it, guys. If you're a fungi, okay, like fungi, right? Like the bread, that's how they make bread, right? Like alcohol fermentation, right? Like you heard that, right? And then like, you know, yeast, right? Remember that, guys? Yeah. 얘네가 미쳤다고. Are they crazy, guys? Why are they doing this without getting anything? Why do they make like alcohol if they're so? The cells are smart. They evolved like billions of years, okay, to be more efficient, right? There's no reason why they run this process without them gaining something, okay? So now, yeah, what are they doing, guys? Why are they running this reduction process? Why? Why do they do that, guys? To what? Recycle this, okay? Which means what? As a coupled process, what happens, guys? Because of the reduction process, what happens? They're running an oxidation process, okay? Oxidate this, turn it back into what? And it turn it back to NAD plus, NH plus, electron. Okay. That's what's going on. That's what's happening, right? So the electron they oxidized is what? Being used to reduce this process. Okay, so you understand what I'm saying? That's the reason why they're doing this. And what? Okay, so I got this back and what? Put this back into what? Step 6. Okay? So truly, you gotta understand, guys, what really holds them back from the process of the glycolysis is what, guys? Yeah, step 6, okay? Which is what? NAD plus and what? Having the glucose available. Any questions? So yeah, so same thing with lactic acid fermentation. So I think lactic acid, guys, I think they changed this guy. They found a mistake, guys. I mean, so this guy, so it's an acid also, right? So you know. So they found a mistake, guys. Lactic acid, like they get rid of this, okay, and then make it into a waste group or something. So that's lactic acid. They found a mistake. Okay? So that's what? That's also a reduction process, okay? With the aldehyde or the ketone into an alcohol, that's also a reduction process. So the same process, same. But again, because lactic acid has 3 carbons, it doesn't produce CO2, okay? I don't think you have to really go that far, but you can kind of get that from the reading, okay? But the core of understanding is what, guys? Yeah, understand what's happening, okay? In terms of, you know, why they're doing this process, okay? So the similar thing happens, guys, by the way. Okay, so down the road, guys, you guys will hear, you know, like the nitrogen fixation, for example, guys. Like, I mean, you guys, yeah. What's nitrogen fixation, guys? Have you guys ever heard of that, guys? Nitrogen fixation, nitrifying bacteria, for example. You guys ever heard of that, guys? Yeah, well, yeah. Why did plants? I don't want to waste my time, but why did plants come to land, guys? Why did plants came to land? You guys like have you guys heard that, guys? The plant was first in water, okay, and then they came to land. Why? I mean, there's many reasons, right? There's many reasons, okay? One of the reasons, one of the reasons is phosphates, okay? They need more phosphates, okay? And also what, guys? They need nitrogen, okay? So they couldn't run like this process all day, and not this process, I'm sorry, the photosynthesis all day water and sunlight, they can produce what? Glucose, right? Problem is what? Problem is what? Problem is minerals, okay, like calcium ions and what? Phosphates, and also what, guys? Nitrate, okay? And now, in the soil of the, land, they have what? We have a prokaryotic cells that runs nitrogen fixation, okay? Which captures the N2 gas in the air while it turns into ammonia, okay? That's the plant uses those, right? Like not just ammonia, but NO3. But why do you think they do that? Like you gotta question it, guys. I guess so. Like okay, so are these bacteria stupid enough to, for plants? We're gonna just donate that to them. Okay, do you think that's really happening, guys, in bio? No. There's a payoff, right? So meaning, guys, that to keep in mind, guys, nitrogen fixation takes a lot of ATP. By the way, they expense their ATP like yeah, how many hoops they have to hop over to get an ATP, but they're using that ATP to run nitrogen fixation. Okay, for the plants, 가져가라고? No way. Just take it? No way. Okay, so you gotta understand, guys, by perspective, guys. So like okay, so you gotta understand the fermentation process. The reason behind this is what? Yeah, recycle this to run it again. Okay, because they can't produce like every time, right? Nucleotide bases, the nucleotide guys here, they can't like produce like every 2 seconds, right? Recycle. Any questions, guys? So, but now, if the cell is a eukaryotic cell that has the mitochondria, then things become a little different story now, right? Yeah, why I gave this equation, guys. There's a question in your homework, guys. Okay, maybe like I'll ask you to do probably the homework is for photosynthesis and respiration together, guys. By the way, but there's a question that so it's kind of funny, guys. There's a question on the exam. I forgot which year, 2011 or something. The question was from the Harvard biochemistry class. Okay, they pulled that question out and put it into the Olympiad biotest, by the way. That uses this. Okay, so you make sure you understand what I'm doing. Okay, like what goes in, what comes out. Overall process-wise. Okay, SAT is 4 sections now?
Yeah.
I thought it was like 7 when I took it. 4 way back. When I took the SAT, guys, it has 7 sections before. Yeah, 7, 8, I don't know, man. Yeah, it's only 4 now. Good for you guys. All right, so what was that, like MCAT? Back in time? I took it 2006, August. This one, MCAT, I went in at 8 o'clock in the morning. I came out at 6 p.m. 10-hour test, guys. So yeah. Compared to this, guys, like, what, this exam, like you were taking a 50-minute test, guys. You got SAT, I don't know how long it is right now. I'm not sure.
3 hours.
3 hours? Still long. Why is it still long?
I don't know.
Okay. Section 1, what's section 1, guys? English?
Reading.
Ah. Oh, glad that I'm not too big, man. Alright, guys, let's talk about, guys, what happens at the pyruvate. So it produces 2 pyruvates, right? So if this is a eukaryotic cell, okay, they do not go into fermentation, okay, unless like situation is not really good. But they store this. They don't use NADH actually, okay. I mean they just save it up, right? So what happens is the pyruvate, if the mitochondria is available, the mitochondria is there, O2 gas is all available and stuff, right? What happens, guys? Pyruvate will go into mitochondria. It will go into mitochondria. Inner portion of mitochondria is called matrix, okay? So those are like SAT stuff, okay? The inner portion. So keep in mind, guys, by the way, mitochondria has 2 layers, 2 membranes, okay? The 2 membranes is mitochondria, by the way, okay? Outer membrane, inner membrane, by the way, okay? So this, I mean, again, like very simple stuff, guys. Inner membrane is called the cristae membrane. Cristae, There's ripples there, guys. They're like ridges. I'm not drawing that, guys, okay? Outer membrane. So why do you think? Because this is one of the questions. Well, this is one of the things that one of my students mentioned last year, okay? So this is one of the things you probably need for your homework as well. Why do they have 2 membranes? So what is 2 membrane, guys? As an organelle perspective, okay, okay, what has 2 membrane? dual membrane like this. There are only two, which is what? 두 개밖에 없어. Mitochondria, chloroplast. But now, why? Why? What do you think? Yeah, why?
Isn't it like one of the origin theories where the larger cell consumes the smaller one?
So why do they have two membranes though? Yeah, what do you think? Yeah, Andy, what do you think?
They fused and like one of the cells like engulfed the one with the mitochondria or chloroplast. So they kept the membrane.
Okay, so what they suggest is mitochondria and chloroplast, once they were what? Once they were a cell of their own, like a prokaryotic cell, right? But they got engulfed, okay? But now, we talked about it, right? When something gets engulfed by the cell, what happens, guys? Like today, what happens, guys? They become like what? A vesicle. 그지? And that vesicle is what, guys? Cell membrane. You know what I'm saying, guys? So, like, dude, like, think about it, right? If this is a cell, that's a mitochondria, okay? Whatever the organelle that was absorbed, okay? And what happens, guys? Yeah, the portion of this will get, this guy will go in, and what happens, guys? And now, what happens, what, guys? Now, whatever they engulfed is what? Have two layers of membrane. Do you know what I'm saying? So that's why they assume, okay? That's what they assume. They don't confirm that what? It potentially has what? Dual membrane. You know what I'm saying, guys? And then the outer membrane is what, guys? Yeah, the outer membrane. This is what? Inner membrane called crispate membrane. Do you know what I'm saying? Okay? That's the, what do they call it? Endomembrane, like the theory or something? That's what they call it, right?
Oh, yeah, it's like the endosymbiosis theory.
Yeah, exactly. Endosymbiosis, exactly, yeah. So this guy, pyruvate they produced as well. So not enough. Okay, so you gotta understand, guys, it produces two NADHs. Okay, so let's look at the equation, guys. Okay, NAD plus H plus turns into what, guys? NADH, right? Yeah. So now take a look at this. Okay, how many electronal balance equation? 2 plus charges, right? Which means what? This one reaction requires what, guys? 2 electrons overall. Do you understand, guys? So what did I say? What do we start from? We start off what, guys? 24 electrons. Okay. Carbon, glucose has what, guys? 24 electrons overall. Okay. And now what's happening, guys, here? So they produced 2 of these guys, which means what, guys? They took out what? 4 electrons, which means what? You got 20 electrons remaining. Okay. So in 2 pyruvates, they still got 20 electrons remaining. Okay? So what are they gonna do, guys? They gotta take everything out. Okay? Where do they take it out, guys? Inside the matrix. Okay, they gotta take every single one of them out. They're stripping all the electrons from the carbon. Okay? They're mugging the carbon completely, guys, okay, in the matrix. So glycolysis, they're nice enough to give them some electrons back, but not in this case. So inside the matrix, guys, the first step, guys, I mean, before, so you guys probably wonder the Krebs cycle, right? Before Krebs cycle, okay? There's some teachers call this like a step. This is not a step. This is a process that occurs before it gets into the before it gets into, guys, the Krebs cycle. By the way, okay? So it's citric acid cycle. So the pyruvic acid. Okay, this is the pyruvic acid. So basically, what happened? What they're doing here is, okay, they're turning this into, okay, before it gets to the Krebs cycle, okay, you probably heard it, right? They turn it into acetyl-CoA. Okay, so acetyl-CoA. So basically, what they're doing is, okay, they're removing this portion, okay, as a CO2. So again, they're oxidizing, okay? So they're getting it out, get the hell out, okay? And then during that process, because it's being further being oxidized, okay, so they still got what, guys? 20 electrons remaining, guys, right? Pyruvates, right? So from here, this process, there are 2 of these guys, right? They're produced, guys, by the way, 2 NADHs again, okay? For the process, and what? Turned into acetyl-CoA. So carbon double bond with the sulfur and CoA. So that's what they're doing. So this is basically sulfur and the acetyl-CoA portion, guys, right there. That's the portion where the reason why you guys this makes this bond relatively unstable, okay, at a high energy. That's why you're doing that. So, but all in all, basically, this is a formation of acetyl-CoA within a matrix of the matrix of the mitochondria, where they're converting the pyruvate or pyruvic acid into acetyl-CoA. Okay, and in turn, they're taking electron out. They took out additional 4 electrons out, so which means they're left with what? 16 now. Okay, 6 electrons still remaining in the acetyl-CoA. And the high-level perspective, guys, that Krebs cycle, okay, is what, guys? 8-step Krebs cycle is what, guys? What do they do, guys? Just take everything out. So now you have 16. So now you have a 16 left, minus 4. You have 16 electrons left, guys. So the Krebs cycle is what? Taking all the electrons out. That's the purpose of the Krebs cycle. So on a high level, guys, what does Krebs cycle do? The next step of this is what? Yeah, take everything out. Take all the electrons out from the carbon and get rid of as well CO2. But this is like a prior to getting into guys, the Krebs cycle, okay? So acetyl-CoA, okay, if you think about it, right? Each of them will have 8 electrons, okay, right now, that they need to pull out. So if you think about it, guys, so the teachers don't talk about it. Like they don't talk about Krebs cycle. What does it do? Like they don't really talk about it too much. Okay, but again, their main job is what, guys? To remove all the electrons from the carbons. Okay. So let's take a look, guys, what Krebs cycle is. So the cycle, and we'll circle. I might not memorize all the enzymes, by the way. So that's something that I need to give to you guys. Okay, like a little piece of paper or something. So oxaloacetate, guys, is where it's kind of starch off, guys, by the way, and then acetyl-CoA. So they're getting acetyl-CoA. So what it turns into, so it's oxaloacetate, acetyl-CoA. So a lot of these guys, by the way, comes up in other stuff actually, like photosynthesis. They use this for a different purpose, by the way. Okay, so it turns into, guys, citrate. So this entire cycle is called a Krebs cycle. Krebs cycle. Sometimes they call this, guys, a citric acid cycle. Okay, because why? Why do you call it citric acid? Because why? Yeah, dude. The first product of the cycle is what? The first, material for the cycle is citric acid. That's why it's called citric acid cycle. I have to kind of look at it, guys, where they pull out the citric cycle. So, because the enzymes, guys, you might want to know the enzymes, but I'll tell you where the important enzyme is, by the way. Okay. So citrate, guys, they turn it into, guys, isocitrate first. Okay. And then isocitrate, guys, they turn into beta keto. If I remember, keto glutarate. Okay, so this is basically here over here. And then keto glutarate will turn that into, guys, succinyl, double C, I think, CoA, succinyl-CoA. And then from here, they'll turn into succinate. And then they'll turn into, guys, fumarate. This was one of your homework, by the way. I don't know if you can recall that. Yeah, this was one of the homework, guys. I think they were asking about like its molar mass or something, and then I don't know. It was one of the homework, guys. Yeah. And then it turns into, guys, a malate. Malate. That's a cycle. Okay. So you might want to know them. Okay. So it's only 8 steps. Step 1, step 2, step 3, step 4, step 5, 6, and 7, and 8. Okay. It's 8 step here. Okay. So I gotta go back and take a look at, guys, okay? What it is, by the way. So I think the enzyme that you guys have to know, I mean, it's better to know all, but I'll send you guys like all the list of enzymes that goes here. I think you have to know the step 5 enzyme, okay? It's called a succinate dehydrogenase. Okay. So this is where, guys, it's turning succinate into fumarate. But I think this is where they produce more NADHs in there. Okay, so, you know. So this is one of the enzymes that you have to know that I pointed out to you guys. Okay. So I think one of the questions, like on the Olympiad, guys, they kind of mentioned, right? If you stop it here, okay, if you stop the reaction here, then okay, so how many NADH do you produce? Okay, so that's what one of the questions they asked actually. So, which means you have to know all the steps here. So let me tell you guys. So NADH will produce one here, one NADH here. Okay, so from the isophytrate to a beta ketoglutarate, that's one. And also here, succinyl-CoA, okay. So this is where they produce also NADH. And then succinyl-CoA to this one, guys, they actually produce here ATP, by the way. Okay. And then from here to here, they actually produce, guys, what we call FADH2, by the way, we'll talk about it, FADH2. And I think the last step, right? Last step produce, this one last step, guys, okay, malate to oxaloacetate produce one NADH as well. Okay. And then I think they remove a CO2 on the, where the CO2 goes out? On these two steps, actually. CO2 goes out here, and CO2 goes out here. Okay. 2 carbons go out on those two. So, like, again, like, dude, like, the reading has it, so they have a better than what I have, by the way, so, you know. So I just wrote it to explain some of the stuff that you have to know, okay? So, first thing first, okay? So, the one thing to know, right, is production of ATP. So, glycolysis can produce ATP, right? And also what? Krebs cycle, which is the citric acid cycle, can produce ATP as well, okay? But now, these ATP, right? How do they produce ATP again, guys? Well, what do they produce this with? With what? A kinase, enzyme called kinase. Every time, kinase. Kinase is the enzyme, okay, in the glycolysis and also Krebs cycle. So it's a different name, like, oh yeah, like, you know, hexokinase, like, you know, there's different enzyme for the kinase, right? But the kinase is there to produce ATP, right? They take the phosphate group out from one group and what? Take it on, put it onto the other guy, right? That's called kinase, right? So, so kinase, guys, by the way, so it's enzyme catalyzed phosphorylation, okay? But now in bio, what do they call this, guys? They call this substrate level phosphorylation. Yeah. So it's called a substrate level phosphorylation. What it means, guys, is what? So phosphorylation is what, guys? Producing what? Like putting ATP on something else. Phosphorylate. Okay. So now the phosphorylation occurs at the substrate level means what, guys? Enzyme catalyzed phosphorylation. Okay. So they use the enzyme, okay, the enzyme kinase to what? Produce ATP. Those are called substrate level phosphorylation, which occurs where? Glycolysis and Krebs cycle. Okay. So as you can see, guys, okay, one of the things is what, guys? NADH, okay, they produce just like what did I say, guys? The acetyl-CoA has 8 electrons remaining to collect. Okay, good enough. So they pulled out, guys, so far 8 electrons overall. Okay, and now they have 16 left. Each of them will have 8 left, right? So if you think about it in terms of electron count, what happens, guys? So one NADH, 2 NADH, okay? And now 3 NADH, there's one more, which is FADH2, okay? This is a little bit different, by the way, but about the same. Their role is the same, okay? It carries electrons as well. So 2, 2, 2, 2, okay, which means that's 8 electrons. So and complete all the electrons we pulled out. So that's basically what Krebs cycle is. That's what they do, okay? Which is what again? Take the electron out and what? Potentially produce well some ATP. So from one cycle, Okay, from the one cycle of the Krebs cycle, what happens, guys? It produces 1, 2, 3 NADH, 1 FADH2, and 1 ATP. Okay? And that's basically the role, and it also produces 2 CO2. So that's basically what you guys have to know, guys, for the Krebs cycle, what it does. Okay. So if the question is asking guys, okay, so if they stop at step 5 of the Krebs cycle, okay, the question can ask you guys, okay, so how many NADHs were produced? Okay, so then I guess you can look out, okay, so like 2 from glycolysis, 2 from the converted to acetyl CoA, okay, 2 each, okay, so total what, 8 NADH. So those are the questions they ask you. So yeah, think about it, guys. If you don't know the steps, you can't solve it. Okay, so there's absolutely no way to solve it, guys, without the steps, knowing the steps for the Krebs cycle or glycolysis. Any questions? Let me know this. Questions, concerns? So now you have to ask yourself, guys, okay? So yeah, so up until now, they produce what? 4 ATPs, that's it. 4 ATPs, okay? So they need a lot more than 4 ATP, by the way, okay? They need a lot more than 4 ATP per glucose molecule, okay? So what are they gonna do now? So let's take a look at it. Okay, let's take a look at, guys, what's happening here. So again, you guys probably all know the next step is basically what they call electron transfer chain, okay? So we're doing this chapter, guys, okay? Maybe 2-3 questions per the exam, okay? So keep that in mind. So, but the effort you have to put in for this chapter, man, like think about it, man. Like the question might be there, might not be there, but you gotta memorize it all. So let's go back to the chart that we had, okay? This is the electron where it's at near carbon, okay? And this is electron. I mean, the oxygen really hasn't played a role anywhere yet. Okay? Oxygen really didn't pop out yet, right? So basically what happens is they use the energy of electron to produce some ATP, okay? So yeah, it went down up to here. Where's electron at right now for us? It's what? It's inside the NADH. So they use some of the energy of the electron to produce some ATP right now. Okay? They use what? They produce 4 ATPs. Okay, so overall. So, so that's due to what, guys? Yeah, maybe some uses the energy of electron, and also what, guys? Some of the energy might be lost due to friction or due to inefficiency, whatever, guys, by the way. But again, the electrons are now at NADH. Okay, and the FADH2, by the way, is a little bit lower. Okay, so this is FADH2. Some electrons right now, I think 4 electrons actually sit here. Some electrons are here, some electrons are there. And what do they do with this now is the question you have to ask yourself. Okay, so what do they do here? So how do they use this energy? Okay, how do they use that? Okay, so how do they use this to mass produce ATP, right? And the answer to that is what? What we call electron transport chain. Electron transfer chain, that's what they call it, okay? So using electron transfer chain, they will start to mass produce ATP. So, so you gotta understand, guys, by the way. So electron transfer chain, okay? You gotta understand what that portion is, and also how does that assist production of ATP, okay? So, so now keep in mind, guys, here, by the way, this is the cristae membrane, okay? Okay, the membrane, inner membrane of the mitochondria. And then, okay, so this one, like I tell you, man, like I haven't seen a lot of questions on this on the Olympiad. Not that I'm not gonna go over this, by the way, but just so you know, like, just so you know, like they really haven't talked about it like much in detail for the ETC portion of it. I'm not sure why. But now they have, okay, so basically I'm gonna explain you guys, by the way, okay, how they do this. This inner portion is called the matrix, okay. And then like the membrane, like the space in between the membrane, guys, that's just called intermembrane space, okay? And they got a bunch of proteins kind of floating around, okay? The first one's called FMN. I'm gonna mention what it is before or later, guys, okay? And here is FAD. And here is U, that's called a ubiquitin, actually. That's one of the lipid, by the way. And then we have these cytochromes again. So this is basically how the ETC, they call this ETC, guys, how it works. Okay, and this is a cytochrome. They call this guy as a cytochrome. Okay, so I'm not gonna write it as cytochrome. This is a B, cytochrome B, and I think C1. This is cytochrome C, cytochrome C. This is cytochrome, I think A and B and A. I forgot, not B, I'm sorry. I think this is A3 and A, if I'm not mistaken. Okay, not that it's really important, guys, right now, but it's I think it's in the reading, maybe not, I'm not sure, I'll let you know. But what this will do, okay? So what these electrons will do now, these electrons are what we call, guys, high energy electrons. Okay, they have energy in there. Okay, right now they have the energy, which is the free energy that can do work. So basically what they're doing is NADH, okay, they're donating the electron to FMN. This is a protein channel on the cristae membrane, okay? So FMN, so if I'm not mistaken, this stands for the flavin mononucleotide, okay? So that's the inner portion of the membrane, by the way, inner portion of the protein, okay? This is a protein, inner portion of it, they have a flavin, like mononucleotide, like, center heme group, by the way. That's why they call it FMN. So you just have to understand this as a flavin, okay, or flavoprotein. That's what they call them. It's nothing fancy, guys, by the way. So basically what it does, the main job is what? They get the high energy electron and what? They pump out H pluses, protons, okay? How many? 4 H pluses, okay? 4 H pluses. Number 4 on there, right? How many electrons? H plus, just pop that. Okay. 4 H pluses, okay? So, and then NADH will turn into NAD plus H plus. Okay, so now they're losing their electrons. Okay, so now they're giving away 2 electrons, right? Keep in mind, guys, here, so now electron will be going into FAD, okay, and then cytochromes, and then basically cytochrome B will push out 4 H pluses as well. I mean, this might be 2, I'm not sure, like 2H2 or 4H plus, guys, doesn't really matter. Electron will go down and then push out 2 H pluses. Okay, I mean 4 H pluses here. So overall it's 8 H plus, by the way. So maybe this is right. So this is what they call, guys, electron transfer chain. So let me explain to you guys. Okay, so it will flow into these proteins. Okay, and they're using the energy of the electrons to pump. So all they do is what? Use the energy electron to what? To actively transport H plus into the intermembrane spacing. Okay. I tell you guys, I haven't yet seen a question where they ask how many H plus they popped out. I haven't seen those questions yet, okay? So the ETC portion of the mitochondria, I'm not saying it might not be on them, guys, but I haven't seen them yet, okay, on the Olympiad questions. So maybe, I don't know, maybe they'll have this year, who knows, okay? But one thing to note, guys, okay, these cytochromes, okay, so if you think about it, guys, these cytochromes, by the way, cytochromes, okay, so a lot of times, guys, when you guys even look at some AP questions, by the way, so like when they're trying to, like when you see some questions about evolution, okay, in AP bio questions, they have a few ways to do so. One of them is what, guys? Like, yeah, compare DNA sequencing, right? Although is what, guys? Compare what? Amino acid sequence of the proteins, right? For different individuals, different like animals, right? So if you think about it, right? So okay, so if they're doing the the amino acid sequencing, which protein are they using? Okay, which is a lot of times what? Cytochrome C. That's what they're using. Okay, so what that means, cytochrome C is very conserved process. So which means for the eukaryotic cell who can perform this electron transfer chain, okay, the cytochrome C, right? It's been one of the protein that actually what, guys? Like, yeah, it's been around for a long time, okay? So if you compare the sequence of the cytochrome C amino acid sequencing, then you can kind of tell me how close I am to the other animals right away. So that's one of the things you probably see. I don't think AP Bio will explain why they use cytochrome C, okay? You don't have to even know what it is, guys, I think, for AP Bio. But cytochrome C are these guys, by the way, where, you know, like it's there, you know, it's a heme group where it passes along the electrons in the electron transfer chain, by the way. So, all in all, guys, so again, one more thing, guys, by the way. So, NADH, guys, they're using all the energy up. So they're using all of the NADHs to what? To produce or pump out H plus from the matrix into the intermembrane spacing. That's what they do, okay? So keep in mind, guys, the FADH2, it's a little bit lower end, okay? Because FADH2, by the way, okay, if you look at this entire process, so NADH goes into the beginning cycle of the electron transfer chain. FADH2, it goes into here. Okay, so the electron is given off here, not on this one. Which means what, guys? They are not giving away 10 H pluses. They're only doing what, like 6. You understand what I'm saying? Okay, so the electrons in the FADH2, they have what? Less energy, because why? They're not utilizing the entire electron transfer chain. So if you think about it, guys, by the way, then where do they produce FADH2, guys? Where do they come out, guys? It goes on what? They come out what? Succinate to what? Fumarate, right? So that's like the step 6, right? Or something, a step 5 or 6, or whatever, okay? So that's what they produce, right? So succinate to what? Fumarate. So that's when they produce FADH2. They're putting right on here, okay? So the FAD, that's one of the protein, right? So, and then what? They do not do this part, but they only do the last 2 portions where they pump out the 6H plus, by the way, okay? So, which means like if there's more FADH2s, what happens, guys? It produces less what? It produces probably less H plus concentration, right? So that's basically, guys, what electron transfer chain is, okay? Where what? They're using all the energy of NADH and FADH2s and what? Pump out the H pluses using all these enzymes, okay? Or proton chains or whatever, proton pump, whatever you call this, okay? And one of the things you have to question yourself as well, what about the O2 gases? O2. And now the O2 will shine, so O2 is what, guys? O2 will pull the electrons, again, turns into what, guys? H2O. So O2, it will turn to H2O, okay, by the way. So now, if you think about this overall, okay, so if you think about it, guys, this right here is FMN, okay, like if you think about it, right, this is FAD. So there is like, you know, like there are these protons, proteins, okay, that will run down, okay. So in order for, okay, this next one to get the electrons and then pass the electron down from the one to the other, what happens, guys? Though every successive proteins should have greater electronegativity. Do you know what I'm saying, guys, here? So, because, like, how is this guy going to strip the electron out from the one guy, okay? So, from the ubiquinon to the next one, which is FAD, or some, I mean, I'm sorry, the cytochrome B and C. How are they going to pull it out? Because why? This guy should have what? Greater electronegativity. It pulls the electron to itself. And that's why it keeps on going down, going down, going down. And what happens, guys? And then what? O2. Because O2 is likely, well, the best one to use, because why? They have very high electronegativity. So as it, you know, this portion is what they call, guys, electron transfer chain, okay? And the chain is possible because why? Every other successive proteins have a greater electronegativity values, okay? And then like FAD, like this guy is what? Like it starts off from here, okay? While this guy starts from here. And the difference of the energy is what? 4H pluses being pumped out. The intermembrane spacing, okay? So what now? Okay, so what the hell now? So this is where, guys, I don't like it's like the way they phrase this, man. I tell you, like, so let's kind of understand what will happen here. So they had glucose, right? So they had a glucose, okay, and they converted this into NADH and FADH2. Okay, that's what they had. Okay, and then what the hell do you have now? And then they use the energy to what? Pump out the H pluses. Okay, Which what is that, guys? What is that, guys? That's what. They call this. So they call this what? The proton motive force. Okay. So, do you guys like? Yeah. Do you guys like physics, right? Proton motive force. Okay. Have you ever heard the term EMF? EMF 들어봤어? EMF? EMF?
Magnetic field.
Magnetic field? So electromotive force, okay? That's called voltage, okay? Like the battery, right? Like battery, you know, like it has like 12-volt battery. Well, that's EMF, okay? Electromotive force, by the way, okay? So yeah, so now it's what? It's not electron, it's what? It's proton here. So yeah, what it means is what, guys? Like what they did is what? With using the energy, they pump all the H plus out here. So high level, guys. Like if you think about it. So this is the matrix, and that's the intermembrane thing. I mean, I just like drew whatever. So they did what? They pump out all the H pluses into here. Okay, the H plus is all pumped out here. Okay, so that produces what? That produces what? The concentration gradient, a higher, much higher concentration gradient of proton H pluses. So like, what do you think, guys? The pH will be a lot lower, right? pH will be much lower. So if I remember correctly, pH will be like around 3 or something. Okay, very low pH. Okay, inside the intermembrane spacing right now. Okay. And then what? I just wondered, like, okay. And yeah, okay, where's my ATP, man? Like, that's the question you have to ask yourself. Like, what was my ATP? Okay, and that's where, guys, the guy named called ATP synthase came along. So there's something we call ATP synthase. Okay. So of course, ATP synthase is what, guys? They produce ATP. that's what the guy does, right? ATP synthase is what? It produces ATP. So yeah, why I keep saying I don't like this, guys? Because this is how they say it, okay? Electron transfer chain, they say, is coupled. They say coupled with ATP synthesis. Okay? So this and this is coupled, okay? By this I'm telling you, if you do the reading, guys, by what, guys? Concentration gradient of H plus, which is a proton motive force. Okay? So what? Like, okay. So they're linked, okay? Or what do I mean? They're what, guys? Coupled by what? Proton motive force. Because they pump out H pluses. Okay? So without that proton motive force, this will not be coupled. It will be uncoupled. Okay? Uncoupler, uncoupled. So first thing first, guys, ATP synthase. ATP synthase, guys, yogo, I tell you guys, the reading that you guys have on the a lot of Campbell stuff, guys, it's not enough for the Olympiad. Okay, like stuff like this, their explanation is very weak. Okay, so not that we have to get like crazy in detail, just let you know, like we don't have to get into like crazy details because I don't think like they're gonna go that crazy, guys. By the way, but the guy who figured out how this works, by the way. He got a Nobel Prize for this, okay? So everything, like, if you do something, Nobel Prize. I mean, I'm not gonna go crazy on this one, guys, okay? So basically, what will happen is, okay, you gotta know a few terms here. So these guys, by the way, so it's one of them, like, so this is basically, on the membrane, this represents, guys, they call this FO portion of the ATP synthase, okay? And the bottom portion is F1, okay, F1, F0, I think. Did I not draw ATP synthase here? I'm gonna swap those up. I don't have the supplements here. The reading is just not enough. Yeah, they don't have the subgroups here actually. Give me a second, guys. I won't give wrong information. Yeah, that's right. Yeah, I have 0 and F1. Yeah, that's fine. Yeah. So, okay. So what this guy will do, because now there's a lot of H plus there. So this creates, guys, you know, the terminology that we talked about in the AP, it's what? Electrochemical gradient, okay? So what it means is that there's a lot more hydrogen, you know, like H pluses there, like the molecule, like the atom themselves, ion themselves, and also what, guys? It has a greater what? Positive charge, okay? And this side is negative. So because all the H plus has been pumped out. So that's what they call, guys, again, electrochemical gradient of H plus, okay? Much higher right now on the intermembrane spacing, okay? Using that concentration gradient, okay? Now think about it, guys. If they open this channel, what do you think will happen? Yeah, what do you think will happen? H plus will what? Come in. You understand what I'm saying? Okay? What do you call this, guys? Facilitated diffusion. Okay? Well, now they put a different name for this. Okay? They call this what? Chemiosmosis. Chemiosmosis. A lot of times they put this chemiosmosis, the term, for H plus coming into going back into a matrix. Okay? During the, you know, electron transfer chain and ATP synthesis. Okay? Chemiosmosis. Chemiosmosis. So basically what will happen is, guys, here now, okay, so as H plus, okay, gushes in, okay, so through the F, it's not F0, it's FO, okay, FO portion of the ATP synthase, what happens is, this guy will start to turn, like a turn body, okay? And now, which will activate the F1 portion. F1 portion has the alpha and beta portion alternating, by the way, so not that we have to really get into right now, guys, okay? We'll activate the F1 process, the alpha and beta portion of the F1 portion of the ATP synthase. And what happens, guys? This guy will be using that portion of the energy when what? Produce ATP. So ADP plus phosphate comes into ATP. So the catalysis the catalysm occurs by the F0, okay? We'll activate the F1 portion of it, and then what? This guy will be able to convert ADP into an ATP, okay? But this occurs at a lot of amount per se. So if I'm not mistaken, guys, one NADH, okay, will produce around 2.5 ATP, okay, and one FADH2 just around 1.5 ATP. Okay, so you get to understand how many can be produced, guys, based on how many NADH and FADH2 produce from glucose molecule. So that's like that's what they call, guys, ATP synthesis, okay? And this overall process, okay, producing ATP, okay, using the ATP synthase, and also electron transfer chain coupled by the proton gradient produced, okay? This entire process is called oxidative phosphorylation. Okay, that's what they call an oxidative phosphorylation, okay? I keep on saying, guys, they will not ask how many ATP will produce, guys. I mean, even for the Olympiad, I don't think for the ETC portion they can ask how many ATP will produce, right? That's one of the not likely questions. If you take the regions, I mean, they ask this question, right? Like, oh yeah, how many ATP produce? Not for these exams, I guess, you know. So I think, what do they say, like overall they produce like 36 ATP or 32 to 36 ATP, guys? It's variable, depends on, depends on, are there's any uncoupler. So that's one of the things you guys have to know, guys, yeah? Which it's not in the reading. I read it, guys, it's not there. I know it's on the Olympiad, by the way. So, yeah. So after all this, right? What do they call, like, so what do you think uncoupler meaning? Uncoupling? Uncoupling this process. What does that mean, guys? What do you think? What does it mean by uncoupling? What do you think? So ATP synthesis, okay, electron transfer chain is coupled by what? The proton motive force produced, okay? So how do you uncouple this process? What do you have to do to uncouple this process? What do you think?
Provide a new source for.
What problem is that? Yeah, how do you uncouple this?
Remove proton motive force
How? How?
by pushing more protons in beforehand.
Okay. Because my logical thing very good. So they say, okay, so uncoupling. Okay, so way to do it is what? So okay, so they produced that, you know, a lot of H plus got pushed out, right? So what do? They just make them, so uncouplers, what do? Instead of H plus coming in through this, they what? Make them leak through the membrane. Okay, that's basically what they call uncoupling of what? ATP synthesis and what? Electron transfer chain. Which in, yeah, what do you think will happen? They will not produce what? Same number of ATP as expected. Okay? Do you know what I'm saying? That's called uncoupling. So what are some uncouplers that you should know? So if I'm not mistaken, guys, okay, one thing that you have to know for the Olympiad, dinitrophenol. Anyone have heard this, guys? Dinitrophenol. Anyone heard this, guys? This, you know where this is found? Yeah, who do you think will need this? Yeah, who do you think will need this? Who wants something like this? Where inefficiency of the electron transfer chain of ATP synthesis. What do you think? Who wants something like this? Yeah, so yeah, let me ask a question, guys. If it's uncoupled, what do you think your body will respond? What do you think? How will the response be? What do you think? So Andy, what do you think your body will do? Okay, if it's not producing sufficient amount of ATP for the cells, then what would it do? As a feedback mechanism perspective. So if you put 1 glucose, it should produce like 36 ATP, right? But it produced like 10. What do you think your body will do?
Does more glycolysis?
Exactly. Yeah, exactly. Run more glucose, burn more glucose. Okay. Now, come on. Where do you think they're gonna put this? What do you think? Where do you think they're gonna put this? Stuff like this, uncouplers of the, you know, ETC. What do you think? Yeah. What do you think? What do you think? Yeah. What do you think? Where do you think they're gonna put this? What do you think? Where? Yeah. Think about it, guys. Like, okay. This is in what, guys? In diet pills. Do you know what I'm saying? So you want to like burn fat, okay? Burn what? Like more energy, right? And what do? Yeah, put this in there. So if you like, what this will do is what, guys? You make your body to burn more glucose. Because what? It's leaking to the membranes. Okay, so it's one of the things, guys, that they call uncoupling of the process. Okay, dinitrophenol. Okay. And the other portion, I think, other part of it also is there's F0 and F1, right? So I think if you deactivate, okay, like F1, if you deactivate F1, so H plus will come in, okay, but if you somehow deactivate the F1 process, what happens, guys? This will not occur. Which means like, okay, so H plus is coming in, okay, but this guy is not active. What happens, guys? Yeah, nothing's gonna happen. That's also like sort of like understanding of uncoupling. Okay. So like uncouplers, like stuff like this, guys, only for the Olympiad. Okay, they will never ask to go to the AP. I mean, but it's good to know. It's that's one of the stuff that you can. I mean, like think about it, guys. If the question is asking which following is an uncoupler, I mean, if you don't know them, then your game is over. Okay, so now, so this is a very like factual, very based on memorization process. Okay, so it's not like applying what you know kind of thing. Okay. Any questions? Questions, guys? Any questions? Let me know, guys. Okay, so guys, I mean, again, the homework that I produced for this one is based on the the photosynthesis also as well, because it's not a lot of questions. If I only give you questions on this one, it'd be like 10 questions, that's it. So I had to combine this one and photosynthesis, okay? Let's talk about, guys, so for the time remaining for now, 15 minutes, guys, let's talk about photosynthesis a little bit. I'm not gonna finish it. But guys, the homework, guys, make sure you guys do your the reading, okay? Keep in mind, guys, if I say, like, if we're doing photosynthesis next week, okay, you guys might want to read photosynthesis like by next week, okay, not like read it like after the class, okay. So, so make sure, guys, have something, know something before coming in, okay. So again, like, keep in mind, guys, make sure you guys read the chapters, and by next week you should be done with the organelles and transport chapter questions. And I'll assign the respiration and photosynthesis. I mean, it's ready, but I just didn't assign. But I can assign that. But you guys have to read the chapters, guys, before doing anything. All right, photosynthesis, guys. This one is photosynthesis, guys, is it's a lot more parts to it. Okay, many more, like a little more going on in comparison to the process we just talked about. So, photosynthesis. So some questions, like some of the the evolution chapters they ask, okay, which one came first? Okay, is it the glycolysis and mitochondria, okay, or is it photosynthesis first? Okay, it's like, is it the respiration, okay, with the Krebs cycles and etc., or is it the photosynthesis? Which one came first? That's what they ask, okay. But a lot of people thinking that because glycolysis, okay, the 10-step process, okay, it only requires like 10 enzymes. So they think because of the it's not simple, okay, it's a lot of stuff going on, but still, this is what, a lot less complicated compared to photosynthesis, okay. So that's the reason why they speculate that this one is the one that came first, okay? And that's the reason why glycolysis is performed by every cell that's out there, okay? So they'll get it because, like, you'll get it, guys, when we start talking about photosynthesis, because it's a lot more complicated process, okay? So let's talk about so we have like 10 minutes left, 15 minutes left, guys, so we gotta be relatively high level, so let me kind of give you an understanding outline of what's happening here. So photosynthesis, guys, by the way, it's what? Like you guys probably heard, guys, it's producing what? Produces glucose. So, and this one can be done, okay, for only what, guys? If you have a presence of chloroplast. So there are very limited amount of organism that can perform photosynthesis. Like, what's the best one that you can think of top of your head, guys? Plants. Okay, plants can perform photosynthesis, and there are some. There actually are some photosynthetic prokaryotic cells as well, okay? Such as, like, some of the bacteria that can be photosynthetic as well. Some of the eukaryotic cells are photosynthetic as well, by the way, but which are not plants, by the way. So, So let's talk about, guys, how they can produce the glucose. Let's take a look at that first. So there's a cycle called Calvin cycle. Let's talk about this one first. High level right now, nothing like too crazy. Calvin cycle. So Calvin cycle, guys. So back in time, they used to call this a dark, dark reaction or something, But it's, Nowadays they call this what? Light independent reaction, okay? Which is basically Calvin cycle. So Calvin cycle, guys. Okay, not gonna draw all the molecules and stuff like that right now, but you understand, guys, what goes in, okay, what comes out. Okay, so high level, guys, here, CO2. You have to put CO2, carbon dioxide. Carbon dioxide was the product, okay? CO2 was a product for the respiration during where? So it's like review, right? Where in the respiration do they get rid of CO2? Krebs cycles and what? Producing acylic-CoA. Acylic-CoA produces production of acylic-CoA from the pyruvate produce one CO2, and also, like, guys, Krebs cycle releases two CO2s, right? That's where you guys breathe out CO2s, right? That's where the CO2 comes from, okay? conversion of what? Pyruvate into acetyl-CoA, okay? Acetyl-CoA into a CO2, okay? So that turns from mitochondria. But now that CO2 is used for the Calvin cycle. So Calvin cycle has what? So like typically, right? It has 3 steps, okay? First one, they call this what? So we call this what, guys? Carbon fixation. They call this what? Carbon fixation, bringing in CO2, right? Yeah, remember I said this in class, guys? What enzyme does this? Yeah, 무슨 enzyme? There's an enzyme that I mentioned in class. I forgot which one is AP or you guys. I'm not sure. I forgot, guys. Which enzyme does this? So remember that I told you guys there's a most abundant enzyme on the surface of the earth, and there's an enzyme that brings in the CO2 for the plant cells. For the CO2, what is that? What was the enzyme? It's called Rubisco. Exactly. There's an enzyme called Rubisco. This is the enzyme that what? Fixes the CO2. Okay, that's the guy who does it. Carbon fixation. Yeah, and then what did I say just like a couple of hours ago? What's the end product of the fixation process, guys? What else do? G3P. Glyceraldehyde 3-phosphate. This is what? This is the product of what? Step 5 of, or step 4 or 5 of what? Glycolysis. That is the product of the Calvin cycle. Okay? And what happens, guys? They can what? Turn this into glucose. Okay? So, yeah. CO2. Yeah, so like you track it back. Okay? Think about it backwards. We turn glucose into G3P during glycolysis. We turn this into a pyruvate. We turn that into what? CO2. So if I go this route, what do we call that? This is oxidation. Okay? It's going this way. Yeah, what is this process? This is what? This is a reduction. You understand what I'm saying? So step 2 is what, guys? Step 2 is what? Reduction. But this is a big problem, guys. Like not a problem, but big question you have to ask yourself. Reduction requires what? Now reduction requires what, guys? They require electrons. Okay? Where are they gonna get it from? Where did they get it from, guys? Electron? Where the hell did they get it from? That's a big question, right? So okay, so I get it. There's reduction, which requires electrons. I need what? Like for 1 G3P, they need what, guys? They need what? 12 electrons, right? Where are they gonna get it from? Is a big question, right? We gotta discuss that. And also, guys, yeah. So if I throw in, okay, electrons into CO2, are they magically turns into G3P, guys? Does it make sense, guys? By turning G3P into CO2, what do they get? They got ATP. They got energy. Okay, so in terms of the energy diagram, okay, this is CO2. Okay, like CO2, like dude, the CO2 is all the way down here. Electron went to carbon. Okay, they're trying to push it back up. Okay, which means what? They require energy. Okay, which means what? You guys, they need. I mean, not only electron, they need what? They need ATP, man. They need energy. Okay, so the big question about, or main understanding, main idea of the photosynthesis is what? What do you think, guys? Where are they going to get the electron and ATP? That's a big question for this chapter. Okay, where are they going to get it? Okay, and how are they going to deliver this to the reaction? That's a question. Okay, now let alone the CO2. They have a problem. Remember I said it, guys? There's a problem with this process also as well, CO2. What's the problem, guys? When temperature gets a little hotter, what happens, guys? They, instead of bringing CO2, they bring the O2 gas in there, okay? So they can't really do, so Rubisco has one job only. They got one job only, they should be doing good at that job, okay? What's the problem, guys? Temperature gets a little hot, what happens, guys? They don't even do this well, okay? They fail to bring a CO2 in. Instead, what happens, guys? They bring O2 gas in there, okay? So the reason is, okay, because why? Like in the atmosphere nowadays we have a lot of O2 gases, but back then when Rubisco was like really just like active, back then, way back, they didn't have that much O2 gas in the atmosphere. That's why it wasn't a problem, but now it is. So the CO2, so again, the fixing of the CO2, like bringing in these material into the Calvin cycle becomes a problem. Okay, so now where are they gonna get it? So high level, right? Indirect source. Where do they get electron from, guys? Every time. Now, if you think about it, guys, if your mom and dad is growing a plant, my mom and dad, they start to grow a lot of plants. I don't know why, man. I have no clue, man. Like, I don't know about your parents. I have no clue, but as people grow older, like I see, like they try to grow a plant. I don't know. They got, if you come by home, guys, you know my parents' house, like dude, they got a lot of these plants nowadays. Like I don't know, but yeah, if you guys have plants, right? What do you guys need to have them survive? What do they need? Yeah, what do they need, Andy?
Water and sunlight.
Exactly. Exactly. Well, right there, water and sunlight. Okay. Yeah. What do you use sunlight for? ATP. Indirect source of energy is what? ATP. Sunlight. ATP is what? Sunlight. Sunlight will provide them ATP. Somehow they should do that. Okay. Somehow they'll produce that. Okay. What about the electron? Water gives them. Water gives them what? Water gives them the electron. Okay. Indirect source. Okay. And that's what they say. Oh yeah. Water will give the electron. Okay. And sunlight will give them the ATP. Okay. So that's the literally, guys, just so you know, the main portion of this entire thing. Okay, so how do they get it? Okay, and what process are they gonna use to get the sunlight converted to ATP? Okay, and what do they do to get the electron out? So basically, guys, where to start? So where to harvest the ATP and electron, guys? They call this process light-dependent reaction. It's called light-dependent reaction. Okay, and the Kelvin cycle in this portion, guys, is called light-independent. Okay, there are a lot of dependency on this one. Okay, so think about it, guys. Light-dependent reaction process. So their main goal is produce ATP, okay, and fix, or not fix, but produce electrons. So electrons, so this happens in chloroplast, guys, by the way. So in mitochondria, electron transportation occurs with the NAD plus, right? But now the difference is, guys, this guy will have NADP plus, okay? So NADP, guys, this is a guy that means the only difference is what? P. This means they got one more phosphate group on them, okay? So again, NADH or NAD, those are what, guys? Those are basically what we call, like nucleotides, okay? But now the difference is what? It's got one more phosphate group on them, okay? So, which means like they got the electron and what turns into NADPH and ATP and deliver it to Kelvin cycle to run this process, okay? So now, so these are the direct source of ATP energy and also the electron. Okay, but now the easy one we can do is basically what we can do is NADPH. So water. This process, guys, is called the photolysis. So photolysis, guys, basically so H2O, as we mentioned, guys, is what source of electron is, right? So H2O turns into, okay. So you've probably seen this on the AP chem equation, by the way, okay? So what do you try to do, guys? You try to convert into O2 gases and H plus an electron. So you've probably seen this equation, guys, okay? This is what's happening. So if I'm not mistaken, okay, this one, so I forgot this is, yeah, so what I'm saying is, guys, this is a not very spontaneous reaction. It will have a negative voltage, okay? It will have an EMF of negative value. If I'm like this, negative 1.23 volts, if I'm not mistaken, okay? So I recall, I don't know if I recall it correctly, negative 1.23 volts. I can look it up, but what does it mean, guys? It will not occur by itself. Yeah. If you have water, okay, does it automatically by itself convert into like O2 gases and H plus and electron, guys? It doesn't happen, guys. No. Which is that, guys? The assist of what? Sunlight. Sunlight is what? Yeah, helping this out. So yeah, that's what they call, guys, is a photolysis. Okay, sunlight, light, helping. Okay, light splitting. Okay, photolysis. So this is what, guys? This is where ultimately the electrons come from. That's where the electron comes from. That's another thing. So that's the indirect source electron, where the electron will be going into NADP, okay, and then this guy will transferring down to that guy and will support the electron for the whole process, okay. So guys, at high level, guys, that's what I mean. We have a lot more to discuss on this one. So probably next week, guys, we'll discuss the homework, okay, and finish off this, and hopefully, maybe time allows, we can go into cell communication actually, okay. So any questions, let me know. Questions, yours? Let me know. So keep in mind, guys, like keep doing your readings, guys. Make sure you guys do your readings, and then get ready to discuss the homework questions, guys, next week. If you want, you can start. I mean, I can assign guys the homework for this chapter, but you gotta do the reading before you do anything, okay? And sometimes, even if you do it, it might not be enough to solve some of the questions on it. I'll assign it when I get home. But yeah, but if you have any questions, guys, please send me an email. All right, guys, and then make sure you guys do the AP homework too. I mean, it seems like AP homework, you guys, those are really easy for some of you guys, I think. So just keep doing it, you know, and then that will definitely help you guys when you guys go to school. Okay. All right, guys, I'll talk to you guys. I'll see you guys on Saturday. Okay.